Hey everyone, we're going to be starting very, very soon, just after 10, but I'm going to give everyone a chance just to sign in, make sure it's all working. And what would be really great is as soon as you log in, if you can, just have a go at typing something in the chat box because I am making this session as interactive as possible because I think that is the best way to make sessions like these as effective as possible. So into the chat box, let's make it a useful comment. Could you let me know, let's see, what exam board are you taking and what A levels are you taking? So which exam board and what are the A levels? Because I'm guessing you're taking biology, because otherwise I don't know why you're here. But biology, I'm guessing, is one of your A levels. What else are you taking as your A levels? And which exam board for A level biology? And I'll have a look in the chats that's coming in. So AQA, biochem and maths. I reckon that's probably going to be the most popular combination because from my experience, it is. If you're taking biology, you're probably taking one other science and or maths. Oh, a couple of you are doing biology, chemistry, and Spanish. I love that. I wish I was better at languages. I always struggled with languages. It's something that I had to put in so much effort to get anywhere. Quite a few of you are taking Spanish. Wow, loads of you are taking Spanish. Is this like a new popular subject? I love it. I've actually just started doing Spanish on Duolingo, so maybe I'll have to see if I can teach biology in Spanish. I won't be able to, but I can dream. Okay, cool. By the way, as well, if you are completely new to me, then you'll soon get to learn my cat B. She absolutely loves it when I go live on things like this. I've got a blurred background, but if you ever wonder what this is, that is my cat trying to steal the limelight. Okay, so mainly AQA and OCR, which I would expect because they are the most common exam boards for A-level biology. But also, if you found me, then I mainly post about AQA and OCR. So that is possibly also why. So it is 10 o'clock. So we're going to get started very, very shortly. So let me just introduce myself and give you a bit of information about how this session is going to work. So first of all, welcome. I am Miss Estruch. If you have never come across me until you just got your GCSE results or even later than that, I have been a science teacher, specifically biology, for I don't know, 16 plus years. I started in 2009. I've taught in schools. In particular, I've done a lot of GCSE and A level teaching, being head of biology, head of science, sixth form tutor. But for the last year, I've only been teaching online through YouTube, social media, and my own basically my own online school. I've got my membership, which I'll talk to you a little bit about later. But I love biology, love teaching, and now I teach thousands of people online. So that's a bit about me. The way these sessions work, I aim for them to be 1 hour. We'll probably do about 50 minutes of content. And in that time, I'm going to be asking you so many questions in the chat box because I find that if you are able to ask me questions and answer questions, you are going to better remember what we go through, better understand it, and to be more engaged. And therefore, hopefully, you'll get the best value for money for your 50p possible. You do get the recording, so if you can't stay for the whole thing, then you will be emailed after the lesson, the recording. You should have already been sent the lesson slides as well. So that's how it works. So let's just go through a couple of other questions that I had for you in the chat box. In this hour, what we are going to go through is review some of the challenging GCSE topics, but specifically, I've picked out two that will feed into your first two lessons. And when I say first two lessons, you might not all be taking the same exam board. In fact, you're not. You've told me that in the chat box already. But what all exam boards tend to have in common is the fundamentals. So they will all teach the same fundamental topics, which typically come at the start of year 12. So no matter what exam board, you'll have one or two of these lessons right near the start. And the information we go through will be applicable. So that's what we'll cover in terms of GCSE. And in particular, I'm going to pick out the key information that you will need to know for A level to help you remember everything you forgot over the summer because you've had a well-deserved, amazingly long summer. but that possibly means you've forgotten everything from May and June. So we'll pick out what you need to know. I'm also going to do a tiny little bit about the change in style of exam questions because you've probably heard a lot that the jump from GCSE up to A level is huge. And that's partly because you now will have more biology lessons per week. So you'll cover more information a week. So it's more to remember. But it's also the information is trickier. You're expected to do more work outside of lessons, but the exam questions can be trickier as well. So that's what we're going to have a look at. And then the last thing is we're going to look at the first two Year 12 topics. So that's what you've got in store. I've already asked you what A-level subjects you're taking, so I've had that information. But back to the chat box, could you just let me know what are you looking forward to in A-level biology? So basically, like, why did you pick it? What are you most looking forward to about A-level biology? So variety in content. Got the exam questions coming in because maybe some people came a little bit later. Homeostasis, so an exam question or topic in particular. Studying anatomy, practicals, human body, anatomy, more depth than GCSE. Oh, you will love A level for that because I've always found that quite frustrating teaching GCSE that I have to give you like a tiny snippet of what's actually happening. So that means sometimes it's just not as interesting or it doesn't fully make sense. So A level, you get a much fuller explanation. So it's more interesting and it makes more sense. Okay, so lots of you, it sounds like you're interested in the human side of things as well. You'll get a lot of that probably about halfway through year 12. So I did introduce myself, but as I think quite a few of you are new to me, because I think almost everyone who signed up to this session found me through the Facebook ads I did, because I don't normally do Facebook ads, but I suddenly thought, I don't think many GCSE students know me. It's most people find me during A levels. So I did some Facebook ads for the first time for this. But if you aren't familiar with me, I've given you a little bit about my background, but obviously, I'm going to say what I do, I think is really valuable and helpful. But here are some students' feedback as well. So they've found these sessions and my videos in particular on YouTube. They've learned so much in biology. They improve loads. Students have gone up loads. And I actually have got at the end of this session as well some of the more recent testimonials I got. Because you might have seen the post that I did. I had lots of students message me this year saying they got D's and E's in year 12 and then they found me during their year 12 revision. And then from year 12 to year 13, they went up to A's and A stars. Now, obviously, that's not all down to me. It would have been changes they've made, but they did say that the videos, the lessons they learned, and the live lessons I did really helped their confidence and understand what they needed to be doing. Okay, so let's jump into then GCSE topics. The two that we're going to cover because they feed into the first two topics in year 12 are cell structure, which will probably take you all the way back to year 9 because you did that at the start of GCSE, and biological molecules. Now, you didn't have a topic called biological molecules at GCSE, and in fact, you didn't really cover much at all to do with biological molecules. You would have learned a bit about DNA structure, you would have learned a tiny bit about proteins, and you didn't really learn about carbohydrates, but you did learn how they were digested. So those are your two topics that you'll get at the beginning, cells and also biological molecules, which basically means the chemicals that are inside of your cells. So you'll be learning things like water, DNA, carbohydrates, proteins, lipids. But in terms of GCSE then, my first question to you is we're going to recap cells. We are just going to do this question here to start with, which is listing the cell structures you can remember. So I'm going to go back to the chat box, listing the cells that you can remember. While you're listing those, I'm just going to have a little look in the chat because I think there might have been some questions that came in that I missed, which, by the way, if you have questions as we go through this, please put them in the chat because I'm here to answer your questions as well. I'll try and mainly focus on the ones relevant to the session though, if we get loads. So someone asked, what do I prefer, humans or plants, and why? My favorite is actually, people always ask me my favorite, and then I list like the longest list of favorites. But in general, it's biochemistry is my favorite, which means anything you learn in biology that is smaller than a cell. So I love learning about immunology, so your immune system. I love inheritance. I love genetics. I love photosynthesis. I love respiration. So I like all the things that are on the small scale, which is biochemistry. Anything that is bigger than a cell, so things like the digestive system, the heart, ecosystems, that is more pure biology. So it's not really plants or animals for me, it's the biochemistry I love. But I do love it all. Right, let's have a look at these organelles. Whoa, loads and loads here that you remember. Brilliant. Okay, so, and a lot of you have got all of the organelles that you need to know. So I can see we've got cytoplasm, cell membrane, cell wall, vacuole, nucleus, ribosomes, mitochondria. Some of you have actually got some that weren't on the GCSE but are on the A level because I saw a Golgi apparatus in there, or chloroplast as well. I don't think I mentioned that. So yeah, that is all of the plant and animals. Next, then, which of those organelles, which is actually the term used for cell structures, when you get to A level, we call them organelles, not cell structures. So which organelles are only found in plants? So there are three from the ones that you learnt at GCSE that you only find in plant cells. So I can see cell wall, chloroplast, vacuole. Perfect. It's those three: chloroplast, permanent vacuole, and a cell wall. Next is, which cell structure is involved in making proteins? So where are proteins made? So inside of a cell, where are proteins made? Now at GCSE, there was one correct answer. At A level, there are two correct answers for this. Oh, and some of you have got both of those correct answers already, which I wonder if that means you've already done a little bit of prep for A level. So it's ribosomes, but the second structure that you'll learn at A level is called the rough endoplasmic reticulum, or RER, and they're rough because they've got ribosomes on them. Right, next, where does aerobic respiration occur? So where in the cell does aerobic respiration happen? Which organelle? Yeah, good. Mitochondria. And then next one, what controls what can exit and enter a cell? That's our last recap from the GCSE content for eukaryotic cells. And which cell structure or organelle controls what can enter and exit? Yeah, so cell membrane. All cells have a cell membrane, and if something doesn't, then it's not classed as a cell. That's one of the reasons why viruses are not classed as cells. They're acellular, meaning they're not a cell, because they don't have a cell membrane. And you'll learn that in A-level biology. So cell membrane, love the extra detail that some of you are putting in. If you don't know that extra detail yet, by the way, do not panic. There is zero expectation that students have gone ahead and learned anything from A level yet. This in itself, you are probably going above and beyond what people are doing. So don't panic if you only know the GCSE stuff. But yes, cell membranes are made up of a type of lipid, a lipid, which is a phospholipid bilayer. You'll learn that later on in year 12. Okay, and then the thing linked to biological molecules. Carbohydrate digestion, because carbohydrates is one of the biological molecules that you're going to learn at the start, and that I'm actually going to teach you today. The only thing you learned about carbohydrates is about their digestion, though. So, where are carbohydrates digested? So, think about your entire digestive system. Where are carbohydrates digested? I'm going to be intrigued to see maybe the different number of answers people give here and the specificity. Okay. So mouth is correct. In terms of where it is digested, it is in the mouth. Some of you have said saliva and salivary glands. Now, they produce the enzyme that digests starch, but no digestion actually happens in the salivary glands. So salivary glands are around here. They'll be secreting the enzyme into the mouth, and then the digestion happens in the mouth. So mouth is number one. The second location is the small intestines. So you then have more enzymes being secreted from the pancreas into the small intestines, but no digestion happens in the pancreas because food doesn't flow into there. So it's the mouth and the small intestines are the two locations. You do actually learn this in a lot more detail, probably in about, I reckon, March of year 12 when you get on to digestion. In more detail, yes, some of you have actually named the exact parts of the small intestines. The very top part of the small intestines is your duodenum. Didn't need to know that at GCSE. And then the lower part of your small intestines is the ileum. You do learn that level of detail at A level. Okay, next question then is, and some of you might have been answering this actually when I did the first question, but the second question is, where are the enzymes that digest starch created? So where are those enzymes created that digest starch? Pancreas, yep, excellent. Pancreas is one. Salivary glands is another. And I like that. We've got extra detail. Salivary glands are producing amylase. Brilliant. Salivary glands and the pancreas. Awesome. And then lastly, what is starch digested into? Also, never apologize for getting it wrong. Learning means getting things wrong. So never ever, because I do so many live lessons. And my key thing is I want people to take part as much as possible. There is never any judgment. People get things wrong. That's how you learn. So don't worry if you get things wrong. So it just means whatever you got wrong, you've now learned that in this session or remembered in the session. So that's always a good thing, getting things wrong. Right, simple sugars. There's different answers that you could get for this. Yes, so starch does get digested into simple sugars. In more detail, though, we get starch, then maltose, then glucose. Different exam boards, you would have learned different levels of detail at GCSE. So simple sugars is sufficient, but it is glucose. Right, so there's a bit of a recap, but I did want to give you this list if you wanted to know what you could do. Sorry, I got my cat here just whipping me in the face. What you could do in terms of looking over your old GCSE notes to prepare. These are, I'd say, the four most helpful ones to review. So cell structure, we've done a bit of that already now, though. The eukaryotic cells, we've recapped a small amount of that, looking at what are the cell structures, organelles, which are in plants only, and some of the functions of those. But if you just look over them and make sure for the eukaryotic and prokaryotic cells, you are comfortable with remembering the different cell structure names and which are in which type of cell, you'll be really sorted for the first lesson. Mitosis and the cell cycle, that won't come up immediately, but you'll probably learn that in November time, October, November time. So that might be one just to start looking over in a little bit of time. Monomers and polymers, that will be your first lesson probably. And you did a tiny bit of that in biology, but more so in GCSE chemistry. But you just need to know what is a monomer, what is a polymer, and then you'll learn some examples of that in A-level biology. And DNA structure, that comes up around December time, but what you would have learned at GCSE comes up again at A-level, but you add in more detail. So there's just a list of 4 things. If you want to know my top recommendations of which GCSE topics to make sure that you remember, it's those 4 for now. Right, so I'm now going to move into a little bit about how A-level is different to GCSE, which, by the way, congratulations everyone on your GCSEs, completing them, getting your GCSE results, and clearly getting on to A-level biology. That is a huge achievement. I hope you are all celebrating your hard work last week. But we are now getting very close to the reality of starting A-level biology. And I'd say the big things that make the jump feel hard are, I've talked about it a little bit at the start, but number one, the pace. You will probably have 4 or 5 biology lessons per week Probably 4. And most schools have 2 different teachers. So you'll probably have 2 hours from one teacher, 2 hours from another. And that is a lot more than you would have had at GCSE. At GCSE, you might have had 1 or 2 hours per week. So because there is more lesson time, you're learning information much quicker. So that's one reason why it's harder, because there's just a lot of information coming your way in a much quicker space of time. So there's more to keep on top of. The second reason is, and it's kind of linked to that first one, there is a massive expectation now that you are more independent, and you will be set homework by your teachers, but you are also expected to independently, without being told, to do work by yourself. And that might be, let's say you have had 4 lessons that week. The work that you're meant to do independently by yourself is going over those lessons to make sure that you remember them before you start the next week. And you might have seen the YouTube video that I posted last week about this. The homework your teacher's set is probably going to help you to go over that content. But if you want to know, well, what should I be doing to review this content or consolidate, that word gets thrown around loads. The sorts of things I recommend are turning those class notes into a small set of flashcards, or I do have flashcards ready made, mark scheme specific, written and edited by OCR and AQA examiners. So if you don't want to make your own, you've got that option, but making your own is actually quite helpful. So make those and then you could test yourself. Or it could be your lesson notes, you want to just turn it into a mind map. Or it might be that you're going to blurt the information, which is where whatever you covered in the lesson, cover it up, write down everything you can remember for 5 minutes, and then check your notes, fill in the blanks. What I would say you should not be doing is rewriting your notes. It is a huge waste of time. So your lesson notes, don't bother rewriting them. It will take you hours and it will not improve your biology. Writing notes again, literally just copying them, but making them neater, prettier, it literally takes hours and it doesn't improve your understanding. It's passive, meaning you don't have to think about it, you're just writing, and it doesn't improve your memory or exam technique. So if that's something you used to do, please don't ever do it again. You could, if you like to have a set of notes, you could use your notes, textbook, and just turn it into a bullet point list or a mind map, but don't bother rewriting notes. It takes hours and it's a waste of time. So those things I'd say. And then lastly is the exam questions are harder. And I can see some of you are saying, where can you find practice questions for A-level biology? I've got on my website for free. If I just find, I'll put it in the chat box now. I've got a range of questions which are arranged by topic and also arranged by skill. So like application skill, long answer skill, math skill, practical skill. And coming very, very soon, I will also have every single past paper, mark scheme, examiner's report, and a video of me answering those questions. So those are going to be on my freebie page. So you can find all of those there. But if I get back to my point, I've gone off on a tangent here. Application questions. This is one of the other reasons why A-level is harder. You probably are familiar with the concept of application questions from GCSE because you had them at GCSE and they're the harder ones. These are the ones where you get a question, linked information you didn't directly learn. So maybe an unknown experiment or an unknown disease, an unknown enzyme, but you have to apply something you have learned to explain that. And at GCSE, you had application questions. At A levels, out of interest, do you know what percentage of your A level will be application questions? Do we know the percentage? And different exam boards, it will slightly vary, but on the whole, it's fairly similar. So I'm going to look out to see what percentage is. 85%. You'll be pleased to know it's not 85%. Okay, and it does actually vary paper to paper. So on average, you're looking at about 45%, 46%. But one of the papers, and this is for AQA specifically now, paper 2, which is the year 13 content, will be 56%. But basically it means almost half of your A level will be application questions. And this is normally the number one reason why when year 12 start and they do their first class test, they might have been getting 7, 8s, and 9s at GCSE, and then they're shocked they get a C or a D in their first A-level test. And it might be they did the wrong revision. They wrote notes rather than flashcards and exam questions. Or it might be they didn't do enough application question practice because they didn't realise almost half of the questions would be application. So let me just go through with you the top strategies of how to revise application questions and then how to answer them. So number one, how to revise them, how to prepare for application questions. An application question, you are applying your knowledge to an unknown scenario. So that means you have to have the knowledge there in the first place. So my tip number one is you need to secure your subject knowledge. So doing things like I just said, using flashcards to check you know the key terms, the key processes, or blurting the information down so that you can remember all the information. But you need to make sure you've done what I call active recall, which means you've actively tested your brain. Can you remember it? Not just watching videos or just reading books, just writing notes. That's passive. You're not testing yourself at all to see, did you actually remember it? So test yourself with flashcards, blurting, those sorts of things. And then step two to get better at application questions is practice. And this is where I gave you that link in the chat box to my website, the freebies. If you were to download the skills assessment bundle, it's completely free. One of the packs of questions in there is called application questions. So it's a mixture of topics, but they are all application questions. It will be tricky for you to complete them all at this stage though, because it covers all the topics and you won't have covered all the topics yet. I do have a few videos on YouTube though, where it is linked to specific year 12 topics. If you search the topic, application, Miss Estruch, and I've got examples there as well. So then that's how you prepare and get better at them. These are my 4 tips though of when you're actually doing the exam question, how to get full marks on an application question. Number 1, you need to read all the information and work out which topic or practical it links to. So let's say you've got an unknown disease. They've talked about how it's has someone got the disease and the symptoms, and you might realize, oh, it's to do with mutations and inheritance. So you now know the 2 topics. Then you'd need to think, right, from my flashcards that I did, what were the key definitions and key terms that come up for mutations and inheritance? And this I'd all be doing in my head. Then you need to think, those key words that I've just remembered, how do I fit those into my answer? Because for certain topics, there are standard marks that always come up. And that's the sort of thing I talk about in my YouTube videos. I have in my A-level notes and I've got on my flashcards. These are the standard marking points, which I said, those are the ones written and checked by examiners. So then we've got number 3, use keywords. Number 4 is sometimes in the question, they might have said, use the information to answer the question as well. So that might mean you have to pick out some data from a graph or use some of the information from the question in your answer as well. So those are your 4 steps. Work out the topic, remember the key terms, and this is where you have to have that knowledge. Use the key terms in your answer, use the information. So those are the key things for getting full marks on application questions, the strategy. I do lots more in terms of exam technique, various things like through YouTube, Instagram, TikTok. I answer questions, I model this, but I'll be showing other things later as well. But now let's actually get into teaching you the first 2 topics. I'm going to quickly just jump over to the chat box before I start though, just in case I've missed any questions. Do the essay questions at the end of the paper count as application? Now that question is specific to AQA. AQA has an essay at the back of paper 3. That is application, yes. So that does count as application as well. Please don't panic about the prospect of an essay if you're doing AQA. I would say the essay is possibly my specialism that I teach, and I do so much for free on YouTube about the essay. I personally love it, and it's my mission to make everyone else love the essay. So if you are doing AQA, the essay is amazing. Don't let it worry you, please. Right, let me just see. I am going to do questions at the end. So if you haven't typed a question already, please don't type one now. I was just checking if I've missed any as I went. Is there also general knowledge question in biology? Not general knowledge. It has to be on the specification. What are the general topics assessed in A-level paper one? So that sort of thing. Have a look at your specification and it will tell you which topics come up on every paper because it's different for every exam board. Okay, right, let's jump into it. So A-level head start. What I'm going through now is relevant for all exam boards. And for all exam boards, you learn it in year 12 and it would be in your first A-level paper, which you would do in year 13. If you are sitting the AS exam as well, then you'll be doing it in the summer of year 12. So the first topic I'm going to go through is carbohydrates, which is usually the first taught topic in biological molecules. Here is an overview of carbohydrates. Carbohydrates, they're a biological molecule, meaning it's a chemical you find in the cells. They contain three elements: carbon, hydrogen, and oxygen. And this links to monomers and polymers. Monomers, if we think back to GCSE, it is a single unit that can bond together to make a larger molecule, a polymer. And a polymer is multiple monomers bonded together. Carbohydrates, you get monomers and polymers. You also get something called dimers. You don't actually have to know that term though, but I've put it in just for consistency. But what you do need to know is these names. They're pretty lengthy, so I'm going to start breaking them down just to make it easier to remember. So the monomer of a carbohydrate is called a monosaccharide. Mono means one. Wherever you see mono, it means one. So saccharide means sugar. So monosaccharide means a single sugar unit, which I'm representing here with that single hexagon. You then have disaccharides, and this is when you have two sugar units bonding together. Di means two. So two sugars, and we can see here, we've now got two hexagons. That's two sugar units joined together. And then lastly, we have our polysaccharides. That is the name of the polymer for carbohydrates. Poly means many sugar units bonded together. Now, technically, it means more than two, because if it was one, it would be a monosaccharide. If it was two, it'd be a disaccharide. Anything more than two, it's a polymer. In reality, it's probably thousands of sugar units joined together. So that's the first thing you need to know, what these names are. So monosaccharide, disaccharide, polysaccharide. And you need to know these examples. So underneath each one, we've got the examples that you'll learn. Monosaccharides, you will learn glucose, that's just a single sugar unit. You'll also learn fructose and galactose. You don't learn much about the bottom two, just that they are monosaccharides. But you do learn about glucose, but I'm going to teach you what you'll need to know. Disaccharides, there are three examples you need to know, and you're possibly familiar with these names already because they came up a little bit at GCSE. You didn't need to know much about them. So you'll learn about sucrose, maltose, and lactose. The level of detail you'll need to know is which two monosaccharides they're made of. And again, I'm going to give you that information. Lastly is the polysaccharides, and these three you should be familiar with from GCSE, but not in lots of detail. So you did learn a little bit about starch, cellulose, and glycogen at GCSE, which out of interest, from our polysaccharides, which are found in plants? Starch, cellulose, glycogen, which of those are found in plants? So those three polysaccharides, which are found in plants. Cellulose, yeah, so cellulose is in the plant cell wall. And starch, yeah, starch is a store of glucose, store of sugar, store of glucose. So glycogen is the only one in animals. That's a store of glucose in animals. The other two are in plants. So that is an overview of carbohydrates. That's the sort of thing that where I was saying don't rewrite your notes. You could make a mind map or something. This information covers probably your first 3 to 4 lessons. I've summarized an overview of it on one slide, but you'll have one lesson introducing, then one on monosaccharides, one on disaccharides, one on polysaccharides. But this is the sort of thing you do as like a revision thing for your wall to help you to remember. Okay, let's go into more detail on the monosaccharides. Again, I've presented this in a way to show you how you could maybe make revision notes rather than just rewriting your notes. So it's more like a mind map. What you need to know about the monosaccharides is a definition of what they are, the examples. Those two boxes we've actually covered already, but we're going to check it. But then I said you need to know glucose in more detail. And what you need to know is that there's two isomers of glucose, and you need to be able to draw them. The level of detail that you have to draw them in varies from exam board to exam board. I'm going to show you the AQA version. You might have to know it in more detail for other exam boards. The way to know this is look at your specification and it will have the diagram in there. But I just want to check isomers. This is actually chemistry GCSE. What is an isomer? Oh my gosh, someone has actually already answered this question before I asked it. It's almost like they can now predict what my questions are going to be. So, what is an isomer? So I can see. Yeah, did in GCSE chemistry, exactly. There's quite a lot of chemistry in biological molecules. It's the biochemistry element. So yeah, it's the same molecular formula, but a different structural formula, meaning they're arranged differently. So you have the same atoms, but they're positioned differently. For GCSE, you needed to know the chemical molecular formula for glucose. What is it? So, what is the molecular formula for glucose? Some of you are asking, is it the same as an isotope? It's not, but wherever you see ISO in science, that means same. So isotope, isomer, you're going to learn the term isotonic. None of them mean the same thing, but they all start with iso because iso in biology means same. So it means same something. Isomer means same molecular formula, but different structure. Okay, C6H12O6. Amazing. Well remembered, everyone. So yes, C6H12O6. So the definition of a monomer, or monosaccharide more specifically, is these are the monomers of carbohydrates. That's what a monosaccharide is. It's the monomer of carbohydrates that could bond together to make a polysaccharide. The three examples that we said you need to know are glucose, galactose, fructose. There are two isomers, and they are called alpha glucose and beta glucose. This is the level of detail you need to be able to draw them in. So as you can see, we aren't drawing all 6 carbon atoms, all 12 hydrogen atoms, and all 6 oxygen. You only have to know it's a hexagon in shape. And then what spikes out either side of our hexagon, and a couple of other extra details. So always start off by drawing a hexagon, and then oxygen is in the top right. You need one branch coming off the top, and then other than that, it's the 2 spikes either side. And alpha glucose is symmetrical in terms of what's spiking off. You have hydrogen on the top and hydroxyl, that's what we call an OH group, hydroxyl, on the bottom. That is the level of detail for AQA to draw alpha glucose. Hexagon, oxygen and a branch, and then it's hydrogens and hydroxyls on the bottom. This is the sort of thing to try and remember. Blurting is great. If you just had, right, I need to draw alpha glucose, draw it. Can you remember? If you can't, you fill in a bright colour pen what you couldn't remember. Try again the next day and keep trying it daily until you can remember it. And then eventually it'll go into your long-term memory. Beta glucose is identical except on this one here, the hydroxyl is now on top, hydrogen's on the bottom. I nearly actually said on carbon one, but then I realized I've not explained to you what that would mean. You don't have to know where the carbons are, or you don't have to draw them in, I should say, but every angle, let's say, in your hexagon is where there's a carbon. So you have a carbon here, 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 and you have one branching out of the top. And we always number them clockwise from the oxygen. So we'd describe this one as carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, carbon 6. You wouldn't have to draw that on the diagram. The reason I say that though is you could, if you had an exam question saying what is the structural difference between alpha glucose and beta glucose, what you would then say is on carbon 1, for alpha glucose, hydrogen's on top, hydroxyl's on the bottom. For beta glucose, on carbon 1, hydroxyl's on the top, hydrogen's on the bottom. So we basically number the carbons. So if you're describing the position, it's more accurate a description. So that is our monosaccharides ticked off. That would be an entire hour lesson for you in A-level biology. Now moving on to your second lesson for biology, disaccharides. The key facts are a disaccharide, di means two. You've now got two monosaccharides bonded together. The name of the bond is glycosidic bond. In carbohydrates, all of the bonds are glycosidic bonds. So that's a new thing for you to learn now. Glycosidic bonds, that is the name of this chemical bond here holding the two together. Disaccharides are formed by a reaction that is called a condensation reaction. So there's our two new key things here. Disaccharide, we'd already said, means two, and it's two sugar units, two monosaccharides. They're joined together by a glycosidic bond, and the name of the reaction that does this is condensation reactions. So that could be two flashcards. Name the bond, glycosidic, name the reaction, condensation. Another flashcard, though, is knowing what is a condensation reaction, because this reaction will come up over and over and over again in biological molecules, because it's the name of the reaction and type of reaction that basically joins together all smaller molecules into bigger molecules. So everything you're going to learn in biological molecules almost. And it's a 3-mark definition. So rather than writing it as a paragraph, I've separated into the 3 bullet points for you. That would be the marking points you'd have to include. That's the sort of thing I do on my flashcards as well. Split it into the marking points, you know what to learn. So the first thing, if I just get my highlighter function on instead, is joining two molecules together. And they are joined with a chemical bond. And in doing that, water is removed. That's your 3 marks for what a condensation reaction is. And this is me here showing you it on the carbohydrates. So here's our hydroxyl group at the bottom. And we're highlighting where the water is going to be removed from. So H2O, water is taken out here. And that leaves you just with one oxygen. So water is removed, oxygen remains, and then we get a chemical bond forming. So we've joined together 2 molecules. A chemical bond was formed, water was removed. That is what a condensation reaction is. That's how we go from having a monosaccharide to a disaccharide, but also how we get polysaccharides plus loads of other molecules. So the last thing you need to know for disaccharides is the examples. I don't expect you to remember all of this, by the way, because you're getting a lot of information for you in one go, but you get these slides. If you want to turn it into flashcards afterwards, you can, or just look over it before each lesson, just so you feel a bit more ahead. And when you're told it, it's not the first time you're hearing it in the lesson, so it should then sink in a bit better. So we've got 3, maltose, lactose, glucose. What you need to know for these is the word equation. So a few tips to help you remember. All of them have one of the monosaccharides as glucose. Because they're made by a condensation reaction, all of them remove water. So that means water is one of the products. So then all you have to remember is, what is the second monosaccharide for each one? And maltose is 2 glucose molecules. Galactose makes lactose. That one I think is the easiest one to remember because it's got lactose written within the name. And then the final one is fructose is the second one to make sucrose. Those could be three separate flashcards just to test yourself on. Can you remember those word equations? Okay, then the last thing is polysaccharides, which again are made by condensation reactions. There are three examples: starch, glycogen, and cellulose. And I already asked you know where they're found. Now, this here is the summary table of all the information you need to know about polysaccharides, which will probably be in an hour, 1 hour lesson at A-level biology. So actually already on to lesson 3 or 4 here of A-level biology. But the key things that you need to know are the monomer. All of them are made up of glucose, but this is where we go back to that concept of the isomer. I'm going to test you in the chat box. Can you remember what was the difference between alpha and beta glucose? Jeez, that's a chance to get a quick drink. Different structure, yep, good. The position of the hydrogen and the hydroxyl group. Yeah, alpha is below, beta, the hydroxyl is above. Yeah, excellent. Now, because they have different isomers of glucose, that affects where the glycosidic bond can happen. I'm not going to go into loads of detail here because it's going to be overwhelmed. I just wanted to give you this so you've got it for notes you can take with you into that lesson so that can help you. But the position of the glycosidic bond, these numbers here, where it says 1 to 4, 1 to 6, that describes which carbons the bond forms between. So carbon 1 and carbon 4 would make a glycosidic bond, 1 to 4 glycosidic bond. We've then got the function, where you find it, and then a bit more about the structure and how that links to the function. But as I said, this is actually now going into lesson 3 or 4. I wanted to give you that information so that you've got that to use in lesson or to prepare afterwards. But I think if I go into that now, we're going to get into a bit of a overload of information because I'm going to quickly swap over to what you need to know about cells because you'll probably be taught this simultaneously. So I'm going to briefly go over this. There's going to be less new information here because you did actually do quite a bit on cells at GCSE. So eukaryotic cells, you learned about at GCSE. You should hopefully remember, you might not remember, but you did learn at GCSE. Eukaryotic cells are bigger in size than prokaryotic cells. Eukaryotic cells have a nucleus, and that's where their genetic material is found. Most multicellular organisms are eukaryotic cells, so animals, plants, fungi, whereas bacteria are prokaryotic cells. And then we're going to look at how the organelles, the cell structures, vary. So I'm going to skip to the organelles that you need to know. These are the ones that you need to know at A level. And if we just have a look, which you would have known at GCSE. So, yes. Okay, so most of those you knew at GCSE. The ones that are new are Golgi apparatus, lysosomes, and the rough and the smooth endoplasmic reticulum. So we're adding in 3 new cell structures or organelles, but you do need to know the structure of these, which you didn't need to know at GCSE. At GCSE, you just needed to know the function, their job. But now you need to know their structure and how that structure links to their function. So the cell surface membrane, that function I asked you right at the start, we said that controls what can enter or exit a cell. It's found in all cells. Remember I said that's why a virus doesn't count as a cell. And you will learn about the phospholipid bilayer, which I've got a diagram of here. You will learn about that structure and how that controls what can enter and exit a cell. So that's our cell surface membrane. The phospholipid bilayer will be the new bit of information that you're going to add in compared to GCSE. The nucleus you learn quite a bit more about because at GCSE you just draw it as like a circle. You know it's where all the genetic material is. If you did triple science, higher tier, you also would have learned about one of the stages of protein synthesis happening in the nucleus when you make that RNA molecule. So the nucleus does contain all the DNA. It's where you get DNA replication happening, meaning copies of DNA are made. And it's also where you get mRNA made, which is needed to make proteins. The structure, this is where it becomes quite different from GCSE. You don't just draw it as a circle now. In fact, you don't have to draw this level of detail, but you need to know that you've got two membranes around the nucleus, and that makes what's known as the envelope. There are holes in that envelope, which are called the nuclear pores. The liquid or fluid inside is known as nucleoplasm. And you also have a smaller sphere inside of your nucleus called the nucleolus. The mitochondria, site of aerobic respiration, you knew that at GCSE. And instead of saying it releases energy, we say it makes ATP, which is a molecule, one of the biological molecules you'll learn that releases energy. It's also got two membranes and it's got a highly folded inside. So we've got a large surface area for respiration, aerobic respiration to occur. Chloroplast, site of photosynthesis, only found in the plant cells, also got two membranes around it. It's also got these highly folded membranes inside, known as thylakoids, and that is where photosynthesis is going to happen. It's got a fluid-filled center, also where photosynthesis happens, and so it's found in plants. The new organelle, Golgi apparatus and Golgi vesicles. This has so many functions. Basically, you are processing and packaging biological molecules, meaning you are making them so the body can use it. So it's where you're going to be processing carbohydrates and proteins. You can also have, actually, that's the next molecule. It's those two, carbohydrates and proteins. They get modified, processed so that your body can use them. And once they've been modified, they then leave the Golgi apparatus in a vesicle, which means like a small ball. And that can then be transported around the body to where that protein or carbohydrate is needed. Lysosomes are basically sacs, so spheres or sacs, and they contain digestive enzymes. You came across one of these in GCSE when you learned phagocytosis in immunology, because you might remember that the white blood cell surrounds and engulfs the pathogen, and then it destroys the pathogen once it's inside of it. The way that happens is you have lysosomes, which contain enzymes that digest and break down pathogens. So once that white blood cell engulfed it, the lysosome then fuses and releases these enzymes to destroy and digest the pathogen. Ribosome, site of protein synthesis. You did that at GCSE. You don't need to know much about the structure, just that they're very, very small. They have two subunits. It's two sections joined together. And you have different sized ones in eukaryotic and prokaryotic cells. Endoplasmic reticulum will be new to you. You have a smooth version and a rough version. And we can see that here. Smooth here, meaning there's nothing stuck to the outside. Rough here, you do have ribosomes on the outside. The rough endoplasmic reticulum, or RER, those are rough because they're covered in ribosomes. And that's why they're involved in protein synthesis. Smooth endoplasmic reticulum don't have any ribosomes, and that is where your lipids are going to be modified, stored, as well as more carbohydrates. So they're going to be processing and modifying biological molecules. So there's a bit of an overlap here between cells and biological molecules, because the biological molecules you'll then learn which organelle they're either made in or packaged up, ready for your body to use. Cell wall. Now, this provides structural strength. You learned about the cellulose cell wall in plants, but there is also a cell wall in bacteria made up of a nitrogen-containing polysaccharide, murin, and fungi also have a cell wall. Cell vacuole, so this is in the center of plant cells, and it helps to make the cell solid, so have that rigid structure or turgid, and it's filled with liquid, and that's why it pushes it outwards to make that solid structure. So key differences, the last little summary between a prokaryotic cell and the eukaryotic cells are, we've said that prokaryotic cells are much smaller, but also all of those organelles that I listed that have a membrane, which was basically all of them, they will not have. They don't have any organelles that contain membranes. Ribosomes don't have a membrane, so they have ribosomes, they're just smaller. They still have genetic material, but not in the nucleus. It's just found as a circular DNA within the cytoplasm. They do have a cell wall, but it's made up of murin, not cellulose. And then some bacteria have additional structures, so a plasmid, a capsule, and a flagella, which you'll learn about those structures and what they go into. So I'm just going to swap over to the chat box because that was quite a lot of information. But this is why I wanted to give you the slides so that you've got that introduction, you've got the key ideas. So if you want to, before the lessons, turn it into flashcards or turn it into notes at this stage, that'd be fine. Or just print it so you can take it to your lesson. You've got that so that when you then learn this, it's not so overwhelming. You should have had an email with those slides in, and you're going to get another email coming with those in. Check your spam, check your promotions as well. Now, some of you are saying, that is a lot of information and you're scared. Let me just recap that. What I have given you there in terms of information, the carbohydrates, that was probably 4 hours worth of lessons. And what we just did there on cell structure was probably 2 hours. So in half an hour, I've given you an overview of 6 hours worth of content, which is probably your first 2 weeks, maybe 3 weeks of A levels. So you will be doing it at a slower pace. You'll be hearing it for the second time. I've also got all the videos on YouTube already if you want to have more of a head start so you feel more comfortable. Please don't feel worried though, because you are in literally the same situation as every GCSE student who's now starting A level, starting from scratch. It will feel overwhelming for everyone. Your teachers know that, so they are there to support you in these earlier stages. So I'm going to go through questions very, very shortly. The final thing I want to just talk to you about is if you are someone that in the past has used tuition, or you think you might want a bit of extra support and you don't want to pay for one-to-one tuition, I want to make you aware of the Biology Study Club, which is that monthly membership that I mentioned earlier that I run. And here's just some of the testimonials from the students that have been in the club. Now, the Biology Study Club is basically myself and two other teachers. We teach live lessons every week for AQA and OCR A-level biology. So you get live lessons, you get the recording, you also get my flashcards for free, you get my study tracker for free, you get my past paper analysis for free. I set you mock papers, I mark them, we go through the feedback. I do live exam questions with you every single week. You're part of a study community as well. So it's like a chat room, but it's just for students in the club. So it's really supportive. I'm in the chat room as well, so I can answer your questions and help you. There are so many benefits to this club, and it's cheaper than 1 hour of one-to-one tuition. So just to recap what the Biology Study Club has, as a year 12, you would go for the gold membership, platinum's for year 13, and the gold membership includes 2 live lessons a week. So you either will get an AQA or OCR live lesson or the exam questions. You can watch the replay if you can't make it live, or if you want to watch it for revision. You get a load of my resources that you normally have to pay for included in the membership. You get mock papers that we create, mark, and go through the feedback. There's a supportive study community, like I said. And this year, loads of students just said how much it improved their confidence, because in the class, they didn't feel comfortable to ask questions, but in the club they did, or they could send me direct messages and then they could. And you can actually do a £5 trial if you want to see what it's like before you commit to it. There's a £5 7-day trial that you can do. You only get access to the live lessons if you do that, though, none of the resources. But for a year 12 student, it's £35 a month, which is cheaper than 1 hour of one-to-one tuition, but you get 2 live lessons a week. So for the price of one tuition lesson, you actually get 2 live lessons a week, plus my resources and all the other things I said. Oh, actually, I've got it all listed there, everything you get. And if you did want some of the notepad stickers and stuff, I'm giving that away as an add-on as well. So that is the biology study club. I'm just going to leave that up. So if you do want to find out more, there is, I'll just paste it because you might want to find out more or look at that free, not free, the £5 trial for 7 days. But I did say I'd save some time for questions. We are so close to 11, but if you've got questions now, I've missed them if I didn't already answer them. Enter them again, and I'm here to answer any questions that you might have. Can you do zoology at uni with just biology, or do you need chemistry as well? Every university has their own entry requirements. You'd need to look it up. Most of them probably want chemistry as well, but some might be fine with just biology. Right, I've got a really specific theory question there that is way beyond this session to do with year 12. So I'm not going to answer the ones to do with the Calvin cycles. That's going to be beyond what we are doing. If you call the OH group the hydroxyl group, what do you call the H? You just call it hydrogen. Do you recommend your AQA or OCR lessons for Edexcel students? Um, it's tricky because the way that I design it in the biology study club, it is so that it is taught by examiners and teachers, all of the lessons. So we will give you exam tips linked to that exam board. However, OCR is quite similar to Edexcel. So I have had Edexcel students join the club before, but you would have to have that extra layer of work you do, check your specification just to check it against it. Should I make flashcards now or wait till I'm at school? I think it's a good way to consolidate your lessons. So I'd potentially wait till after your lessons, but maybe make some for what you've just gone through now. How exactly would you, oh, I've missed one. Would you draw the structure in an exam, e.g. mitochondria? They don't, I haven't actually ever seen them ask you to draw the structure. So they don't get you to draw it. They give you the diagram and you have to label the structures. Or they might give you a microscope image and you then have to copy the microscope image, but they've never asked you to draw an organelle just from memory. What times will the study club be on? Okay, good question. AQA is Mondays 5:30 till 6:30. In fact, I'm going to need to just double check this. Get my timetable up. So AQA 1 hour lesson is 5:30 till 6:30 p.m. Tuesdays is OCR live lesson, exam questions, 8 till 8:30 p.m. Wednesdays is OCR 1 hour lesson, 6:30 till 7:30 p.m. Wednesdays, 5 till 5:30 p.m. is exam questions. So it's all Monday, Tuesday, Wednesday. There's nothing on a Thursday or Friday, but you get the recording as well if you can't come live. But it's best to come live because then you can take part. Right. Is the skills assessment bundle for OCR2 or just AQA? Those ones are AQA. I do have, where are they? My OCR ones are actually inside of the biology study club. So it is AQA at the moment, the past paper questions on my website. I have an exam of biological molecules and cells on the first lesson back. How would you recommend I revise? As a year 12 student, oh my goodness, that seems extreme. Or maybe you're a year 13 and they're giving you that. What I would say is to revise for that test, watch the videos I've got on YouTube that cover it or read your notes if you've got them. Test yourself with flashcards and then have a go at some exam questions would be best. So someone said they feel a bit overwhelmed by how much we did today. We did do a lot. So A level, you learn this level of detail, but it's over a much, much longer time. So if you remember, I said in that last half an hour, that was probably the first 3 weeks of content. So it would have felt a lot. Your first lesson will just be, if I go back to it, where is it? Your first lesson will probably be this slide only. So you'll just cover monosaccharides, what they are, examples, and drawing it. So that will be what you'll cover in 1 hour. So it'll be far more manageable. And then it'll just be after that lesson, checking you can remember that. Your second lesson will then be disaccharides. So today I've given you an overview of a lot, but bear in mind, most of those slides, one slide, you would have an hour of being taught it, checking you understand it, remember it, and then you've got time after the lesson. So we have covered a lot. You will do it at a much slower pace than today. So um, it will be far more manageable. Um, and yeah, and also bear in mind, everyone will feel that way. It's a lot of new information, but it is at a much slower pace than we did today. Someone says, does a carbohydrate need to always have carbon, hydrogen, oxygen? Yes. By the 2nd of September, if I revise a few hours a day leading to it, would I be better prepared? You would be better prepared, but it's not the expectation. But you might want to just have a look over monosaccharides, disaccharides, and cell structure, just so in the first couple of lessons you feel quite secure. In our lessons, should we make our own notes because my teachers give us OneNote templates? Different teachers will have different expectations. Some might want you to make notes as you go. Some might want you to actually have notes they give you so that you're better able to just annotate, listen, and take part. Just whatever your teacher wants to do, that's what I'd recommend. Um, do you think, oh, that's about university, so I'm not going to answer university questions right now. Miss, because we're going through problems, is when you can give discount. Yes, so with all of my resources, I do what's it called? Discount, the bursary discount. You just need to email me and you get a discount if you're a bursary student. It's hydrox. So someone said, is it hydroxide or hydroxyl? So I'll write spelling hydroxyl group. If we did well in our GCSE and in class before A level, if we did well in our GCSE and in class before, then an A level won't be such a nightmare. Yeah, exactly. If you are someone that is, I mean, put it this way, if you are worried that you might not be good enough, you're probably the kind of student who will be good enough because you clearly care. And A levels is a step up for everyone. And the people that are aware it's a step up and care about that will then put in the work that's needed to make it feel manageable. A levels on the whole do feel hard for everyone for the first month, and it's just accepting it is going to feel hard and you're not alone in that, and realizing it won't feel that hard forever. So if you were to ask a year 13 now, they'll probably say, oh yeah, it was a massive jump. But then if you said to them, oh, what's the carbohydrates topic like for you? They'll be like, oh, that topic's easy. Whereas at the time when they did it, they would have thought this is so hard. There's so much to remember. It's so confusing. But now if you ask them, they'll say that's a really easy topic because it's all relative. When you start year 12, it's all new. It's difficult. It's maybe a change in school. It's a change in routine, change in expectations. So it is hard, but you will get used to it. So it's just accepting at times at the beginning when it feels difficult and hard, just know that is normal and ask for help, follow the guidelines that you're given by your teachers, and it will become more manageable. But just don't ever think you're the only one struggling because you definitely are not the only one struggling. Everyone will find it hard. But then if you do the right things, it will feel more manageable. Let's see. When does the biology study club start? It's already started. It's been running for 2 years. I'm running a lesson tomorrow. The first AQA one this week is tonight. It's already running. So if you signed up for the 5-week taster, you could come to a lesson tonight, basically, and you could come to my one tomorrow. What time or how much time should we spend every day after a lesson looking over topics, and what should we be doing? Most of the time, the homework that you set, if they are setting you regular homework, it's probably sufficient. But if you haven't been set homework that day or week from a lesson, I would recommend maybe spend 20 to 30 minutes. And in that time, that's when I'd maybe make a small number of flashcards and test myself, and that'd probably be enough. What is coming up in tonight's lesson? Good questions. If you did want to come to it. Tonight's one. Oh, it's a year 13 one tonight. It's photosynthesis. So photosynthesis is tonight, and the one tomorrow is year 12, translocation. But it's a topic obviously you've not learnt before. Um, the first year 12 one that is probably going to be really helpful for you is next week because there'll be a carbohydrates lesson and I'm doing carbohydrate exam questions on Tuesday next week. But if you signed up for the trial week today, that will include up until Wednesday next week. But if I just do the reminder, if you are interested, I do really need to emphasize that the price is going up on the 1st of September because the number of lessons is increasing. So the price goes up from that point. But if you sign up now, you will pay £35 forever. It won't ever go up for you unless you quit and came back. But when the prices go up, that price increase is only for people that haven't signed up yet. So if you were to sign up today on the £35 per month, it will never get more than that as a gold member. The platinum membership for year 13s is more money, but that's because they've doubled the quantity of lessons. If I'm going to end it here though, because I've realised it's 10 past and I've been going on and on. But if you've got any questions that are linked to the Biology Study Club, please send me a message on Instagram or you can email me missestruch@gmail.com and I can answer any questions linked to that. I hope you found it helpful and you've got those notes to help you. My big takeaway is please don't feel too overwhelmed. This was a lot of lessons I've covered in that half an hour. Everyone does feel some level of overwhelm or anticipation and finds it hard. That is completely normal. Everyone feels that. Follow the advice from your teachers. Try and look over things when you can. And if you are worried, please ask for help. Those are my big things. Your teachers are there to support you and to help you. They won't be judging you. They are there to help you. So never, ever feel worried about asking for help. That's the best way to improve. Best of luck. And as I said, I've got all of the videos on my YouTube channel for free. But if you've got any questions about the flashcards, the Biology Study Club, just send me a message. I'll be answering those later today, but I'm going to tutor right now, so I can't answer them now. Best of luck, everyone. And I'm sure I'll see you in some form or another soon, whether it's on YouTube, social media, or hopefully I'll see some of you in the study club and I can say hello to you tomorrow or next week. But that is it for today. Best of luck with your start to A-levels, everyone.